Welcome to Alma Connects Know Your Alumni. Hello everyone, I am Tejasvi. Wishing you all a harmonious day both molecularly and mentally. Welcome to another podcast episode brought to you by Alma Connects. This series brings forward some of the most inspiring stories from our alumni who have turned their passion for learning into purpose and progress. And today, I am super excited to introduce a guest who perfectly embodies brilliance and dedication, Miss S. Siksha Sravani. She is currently working as a proposal engineer at Praj High Purity Systems, Mumbai. She completed her B.Tech in Biotechnology from Vignan's University with an exceptional CGPA of 9.83 and her M.Tech from NIT Warangal with 8.81 CGPA. She also qualified GATE with an All India rank of 778 and has gained valuable experience through research at IISER Berhampur and Nanyang Technological University, Singapore. Thank you so much for joining us, ma'am. We are thrilled to have you today.
Thank you so much, Tejasvi. That's a great introduction for me. Hello, everyone, and I am Shiksha Stravani.
So, to begin with, could you take us back to where it all started and what made you to choose biotechnology as your field of study?
Okay, so it's all started with when I was in 12th class. We used to have this chapter or like entire unit which was related to biotechnology, right? So this chapters of biotechnology, principles of biotechnology fascinated me a lot more than being a NEET aspirant. So that moment was something like I wanted to do something related to this field. Then I have Googled, searched it for genetic engineering, tissue engineering, biomedical engineering, and all stuff. And then my father brought up this Vignan University have this great program called Biotech Engineering, and then we happened to join here, and that's how it all started regarding my biotech journey.
And that's funny. You have always been an outstanding student, consistent and driven. What kept you motivated through the ups and downs of your academic life?
Okay, so I was also an average student initially, but when I started studying, each one of the thing has created a curiosity in myself. Like, what is this? Why? When? How? Why it started? Why it's going? So I have started a bit curiosity in myself, and at all the end, that led me to create a concentration over like in a good way and improved my confidence, and that's how it started, and I hopefully it didn't end till now.
Was there anyone or anything at Vignan that particularly influenced your journey? Maybe a professor, project, or experience that shaped your goals?
Of course, Vignan is the foundation for my research journey. So I would tell it by heart that Professor Dr. K. Shrikant and Vijay Ramu sir, they both had laid a great foundation for me. And imagine being an undergraduate student in during my second year itself, they have started a project and also published a paper by my final year, which is a great achievement for any student life. So I would say it proudly that Vignan has given me a great foundation.
That's really inspiring for Vignan students. Your M.Tech project on groundnut derived scaffolds for bone tissue regeneration sounds fascinating.
Yeah.
What inspired that idea and what was the key finding from your research?
So when I talk about groundnut derived scaffolds, so this idea was initially we are working on natural scaffolds because synthetic scaffolds which we are using in tissue engineering, they are very like non-biodegradable and they were causing some kind of immunogenic effects. So in order to rectify these things, we have come upon a product which has a very strong fiber like groundnut shell. So we have thought of fibers from apple and some other things, but we targeted the strongest bone, femur bone, so that it is a strong. Most of the people get injured in that area. So we thought it would be more reliable if we go with groundnut. And the key findings are that we have successfully able to differentiate osteoblast like cells MG63, and we were successfully able to make a scaffold out of groundnut shells.
Going to another question. You have also published a study on pila virens and cigarette toxicity, a very unique environmental angle.
Yeah.
How did you get interested in that area of research?
So this is also one of the fascinating study where this is the project where I have started my foundation. So there is this biochemistry lab going on with my professor Dr. K. Srikant. So he just asked a question in our class. I would ever remember this question all the way, all the time. So he asked us what would be a sex of a earthworm. So guys, you do just imagine what would be a sex of an earthworm. So I told, everybody was thinking so many things. I just whispered the answer sitting in the first row that it is a hermaphrodite. Sir was like, be quiet. And then sir called me and he gave a small assignment and lit and going upon, and we started working upon different assignments, and then he included me in this project, and that's how I developed interest upon the project, and I have learned different techniques like ELISA and also how to incubate with and handle the animal handling things like pilovirins. So I have learned different techniques and how to do antioxidant assays.
That's great, ma'am. You have explored both wet lab work and computational biology. How do you think these two complement each other in modern biotech research?
This is very interesting question. So whatever the work a wet lab work member does, it is obviously has to be done by a dry lab sequencing. For example, if we do a MTT assay, for example, so the for MTT assay the results has to be analyzed via a dry lab technique. Even if you are doing a DNA extraction, to know the sequence, you need to do a NGS sequencing. So both are correlated and both are very important. So if a person knows the both techniques and both and that person can make it more easy for himself to understand and analyze the result what he is working into.
Okay. And if I may ask, was there ever a point during your research when you thought this might not work? How did you handle those moments?
Oh my god, oh my god. There are so many moments that I had thought like, oh my god, this is not going to work. So initially when we are talking about the groundnut shells, so we thought of doing with apple first initially. That was very weak that I couldn't make it. And then I did with groundnut shells. And with groundnut shells, I was unable to get the support to make it very strong, enough like a bone tensile strength. I was like, done, I'm not going to do this. One day I was doing cell culture and then I have got a lot of contamination, which I don't even know what kind of contamination it was. And that was also a moment I used to cry and rant when my experiments get used to fail. But then my professor, Parcha Srinivas Rao sir, used to always motivate me and always made me to be a resilient student.
That's
Yeah.
great, ma'am. You have worked at NTU Singapore and IISCR Berhampur, both impressive experience. How was that exposure there different from what you had in India?
Okay, both are like very unique experiences. Both are the things which I have learned a lot from each of the labs. So there is nothing different I do feel. Or maybe the equipments are different, maybe the lab protocols, the lab environment might be different. So, and maybe the professor's strictness is different. So whatever the place or wherever the place it is, how is the lab place, irrespective of the things, if you are someone who is very curious, patient and dedicated, the lab place doesn't matter. I realized this being in different labs.
That's true, ma'am. Could you share one unforgettable moment from your international research journey, something that truly changed your perspective as a biotechnologist?
Okay, first I used to think, I am someone person who used to think failure is a very bad thing. I shouldn't fail anytime. So I was that person. My perspective of failure has changed during that moment, during my international internship. So this happened, I was doing a protein purification. I've did couple of purifications, I'm not getting good result. Even after getting a result, they are not getting good collagens what we were expecting. I was failed. I couldn't take that. So I used to rant, I used to cry, why I'm not getting this? And then I realized that that was the changing moment that in science it is okay to fail. In science it is okay to repeat. continuously. And in science, it is the final thing is to get discovered something new what is not known. And in science, during that moment, I realized that I love to be in lab and to be a researcher.
That's really motivating, ma'am. You have now transitioned from research to industry as a proposal engineer. That's quite a shift. How did you navigate the transition?
Okay, this transition wasn't actually an easy transition for me because being a wet lab person and being a researcher. So, but I had learned things. I should thank personally my manager team, my team in the Praj Industries for helping me out for learning new things. But yeah, this transition also helped me to learn especially GMP, like good manufacturing practices, CIP, SIP, sterilities of a bioprocess system, how it should be. If I have know how to do a bench work, if I know how that industry is working, I know the both scales, so that I can be the person who can balance the both levels. So that would be something this transition helped me out.
Okay, ma'am. How does your current role connect with what you studied? Do you find your academic background helping you in day to day work?
Yeah, because we in my BTech in Vignan, we in third year we have heat mass transfer and biotechnology, bioprocess subjects. We whatever we have in undergraduation, they help me a lot because the terminology, the process, the equipment, the mechanisms, how they work, the calculations we do, everything are very relatable in my everyday, day to day work life.
Okay. For our listeners who aim to enter the biotech industry, what do you think the most important skills to build early on?
So there is no one particular skill. It's always something you like, not you. It's like anyone, me or you, anyone has to inculcate the things, learn whatever you're getting into. If you're, confidence speaking on stage is a talent, that is also is a talent, that is also is a skill. So developing any skill is the main thing. So if you're talking about particularly into biotech, you have to learn, if you're learning one technique, like for example protein purification, so in one protein purification you're going to learn several things under it, like microbial culture, transformation, large scale, subscale culture, FPLC, chromatographies. So you get to learn different types of skills under one technique. So whatever the thing you learn, it is for wet lab. So when you do a dry lab, when you're doing an in silico study, you learn to know different types of databases how to use, and you learn to use how these interactions are being happening, and you know how to do a docking, how the studies are being done. So for this a wet lab work. So there is no one particular skill. So it's all the way you want to go, the journey you wanted to do, you have to make your route in that way, learn the skills in that way. That's how I can tell for this right now.
Going to another question, you have earned several recognitions from the NPTEL Star Award to Ideathon wins and even organizing major events.
Okay.
Among them all, what's one achievement closest to your heart?
Ideathons, I feel the most achievements, because I have found myself that I'm very creative and also innovative, and I'm found myself to be an entrepreneurship. So thanks to Sandeep sir for identifying such a skill and making me win many awards, the Best Idea Award and several awards for this my idea. Thank you so much. So I would say Ideathons are very best for me.
Yeah, ma'am. And as someone who is being a student leader and an active participant in every opportunity, what does leadership mean to you personally?
So leadership, being a student leader, sometimes it is critical as well. But personally, being a leadership means being empathetic to everyone, balancing everyone, and able to be the first person when a challenge arises. When you get a failure to your team, it's also your failure. We have to take the responsibility why this failure has been happened and why it has to be done and how it should be rectified. That's what a leader should do. And when it is success, an entire team is responsible for the success. And when it is failure, and it is same as well.
Very impressive answer, ma'am. You have been part of research, teaching, and now engineering. What's your mantra for staying adaptable across such diverse fields?
Okay. My mantra would be I just, whatever I do, I enjoy doing it. If I'm doing whatever thing I'm doing it, I try to get satisfaction with the work I do. That's the only mantra. There is no mantra, no nothing like. It's just the satisfaction I get whenever I'm doing something that makes me happy, enjoyable, and like if you're watching a movie, you enjoy it. If I'm teaching, if I'm doing some work in lab, I'm enjoying it. So that's how it is, and I get a lot of satisfaction for that.
Okay. What advice would you give to students who dream of pursuing higher studies or research abroad?
So I would suggest, like, if you're going for higher studies, I would definitely would suggest that you all guys should prepare for GATE. It's a very good exam. People are just ignoring GATE. I feel like you get a very good packages, very good knowledge, exposure. You would be in national institutes, and that is a greater opportunity to be in such kind of institutes and learn more of information and do a great projects and interact with high level of intellectual people. So that's how, if it is for higher studies, I would suggest. If you're going for masters abroad, the same thing applies. And always remember that CGPA are also important, but people do say that CGPA doesn't matter, but to show off your skills, I feel somewhere CGPA matters, because they do scrutinize our resumes, seeing at the first level her score is less, maybe not her. That fear makes me feel that CGPA always matters sometimes.
Yeah. So, what message would you like to give to the young biotechnologists of Vignan?
Oh, again, coming to first thing is maintain CGPA. Don't miss your lab sessions. Learn a lot of lab skills. Develop creativity, curiosity, and definitely patience. If your experiment do fail, don't get upset. We have to be strong and stand up again and do the experiment.
Yeah, ma'am. And moving to our fun segment, it's rapid fire.
Oh.
Your favorite research tool or instrument?
Favorite tool or okay, my cell culture is very fascinating experiment for me. I enjoy doing cell culture. Mostly it might be a subculturing or passaging or trypsinization work.
A scientist or innovator you admire most?
Okay, okay. If when it comes to a person I more admire as a professor, as a scientist, is my professor Pacha Srinivasrao sir. And when it comes to a scientist, like a Nobel laureate, Shinya Yamanaka, he is a person who has introduced iPSCs, induced pluripotent stem cells, which has been a revolutionary thing in tissue engineering and stem cells and regenerative medicines.
The one food you can never get tired of?
Okay, I never get tired of death by chocolate from Creamstone. Guys, it is not advisable to eat it every day, but I eat it to stress myself out.
If not biotechnology, what career would you have chosen?
I would be a teacher, or I would open a painting stall with a with a cafe or something.
That's nice. Your dream travel destination?
My dream travel destination would be Iceland. I would love to see Northern Lights there.
One thing you do when you feel stressed?
Oh, this is the very great thing. My father, I call my father or my friend, just rant for 1 or 2 hours and tell everything what is running in my brain, so that give all my headache to him for 1 hour, and I cry, I rant, I tell things, so that I just keep myself calm and sane, process, and go to the next place and next work very calmly and do the things go smoothly. So my father handles that thing.
A book or movie that inspired you recently?
Okay, I am a book reader. I read the genres related to suspense thrillers and psychological thrillers. I'm not much more of a non-fiction reader, but once I have read this book called Wabi Sabi. So this is one of the amazing book. This is the only book I have read because I have been suggested to read. So this shows how resilient one has to be in their life. Yeah.
Okay. What motivates you every morning?
A cup of chai from my mother motivates me a lot.
A hobby that keeps your creative side alive.
I do paintings a lot. Recently, I have did a lot of paintings sitting at home. So yeah.
And lastly, what's your honest opinion about this podcast experience?
This is very great opportunity. Being from one side of the chair to the other side of the chair is very exciting, very fascinating, and I'm very grateful, thankful. I know I'm repeating these words so many times, but I'm very much excited as well about this podcast. And thank you so much, Vignan University and Alma Connects, and Vice President Navya and President. And Amaya, Tejashwini, thank you so much for giving me such a great opportunity.
Thank you so much, ma'am, for joining us today and sharing your incredible story with such warmth and detail. From being an outstanding student to an accomplished professional, your journey truly reflects the power of consistency, curiosity, and courage. It's been a delight having you here on Know Your Alumni. Wishing you even greater milestones ahead. Thank you, ma'am.
You're welcome. Thank you.